Okay, good morning, friends. In the last three classes, that is on the last Friday, Saturday, and Sunday, in three classes, we discussed about various plant diseases caused by fungi, diseases caused by bacteria, diseases caused by viruses, and also diseases caused by mycoplasma. So we discussed all of them in the last three classes. Apart from that, we also discussed about various pests that attack standing crops, and also they damage, cause a lot of damage to the stored products. We also discussed about angiospermic parasitic plants. So we discussed in the three classes about them. To repeat, we discussed about various fungal diseases, bacterial diseases, viral diseases, and also mycoplasmal diseases, and also various parasitic angiospermic plants that cause a lot of damage to the standing crops. We also discussed about various pests. So generally, when I say pests, generally we concentrate on insects. We concentrate on insects. Insects, they cause a lot of damage. Pests, not only insects, even small animals like rats, bandicoots, and birds, they too cause a lot of damage, not only to the standing crop, but also stored products, especially in the go down areas. According to an estimate, from the field to the consumer, a big chain is there, that is transport or food chain is there. In this food chain, from the crop, that is from the field, from the farmer, it reaches to the market, from there to the mill, then to the go down, and then to the fair price shops, ultimately to the consumer. So in this chain of activities, at each stage, it has been worked that at least 5 to 10% of the produce is being lost. So like that, a lot of damage is being experienced, especially in the third world countries and in the developed countries. But whereas in developed countries such as USA or even in China, the produce or the agricultural products, they have their shelf life. For example, if you take vegetables or fruits, their shelf life is very, very short. During that period, unless you consume, they get spoiled. Once spoiled, we have to throw them. But in developed countries, they have developed a number of, or devised a number of processes wherein food processing is taking place, and by adding various preservatives, they are being preserved and are released to the consumers as and when they are in need. So according to an estimate, so in America, something like around 60% of the produce is being processed and being preserved. Whereas in China, it will be around 23%. But coming to India, very poor percent, just 2.3% of the produce is going to processing. So thereby, so we are losing a lot of produce while transporting or in the food supply chain. So now today we'll concentrate on this disease pest management. So far we discussed about various diseases caused by fungi, bacteria, viruses, and mycoplasma. Now here onwards, in today's class, tomorrow and day after tomorrow, we concentrate on how best we can check the spread of the disease. How can you, avoid a contact between the host and the pathogen? So this will be concentrating in today, tomorrow, and day after tomorrow's class. So here, first part is what is called diseases. Now the second part is management of the diseases. First of all, identification of the disease is a must. So you should know the cause for that particular disease. Unless and until you know this specific pathogen, its life history, its mode of replication, etc., you will not be in a position to control the spread of the disease and check the spread of the disease. So once you identify the problem, looking at a plant, you know whether it is healthy plant or a diseased plant. Of course, diseased means it's physical and it's biological, it's yield, its physical features, and its physiological functions get affected. And generally, the face is the index of mind. So if you are healthy, you look glowing, you look healthy. So also with the plants, if they are healthy, they are very green and very attractive. But if they get infested either by a pathogen or by a pest, generally they show consecutive or the plant, response. The plant response. When a pathogen, when an alien particle enters into any living organism, it shows its response. So during this, there is a tough fight between the pathogen and the host. And this results in the damage of the certain tissues or cells, and they will be visible in the form of what we call symptoms. So by looking at the symptoms, you will come to a conclusion to which is the pathogen responsible for that symptom. And once you identify the reason for that unhealthiness or the disease of the plant, after knowing that, then you will start curative measures. So here, the disease or pest management involves identification of the organism and the identification of the disease. Then how best you can check the spread of the disease, how best you can protect your crop, or how best you can protect your stored produce. So now today, what we do is we will learn about various concepts of and principles of pest and disease management practices. Generally, here the pest management. So what do you mean by this pest management? So pest includes both pathogens or the algae, fungi, fungi, bacteria, your mycoplasma, viruses, and also pests. Pests, I mean various insects. The leafhoppers are there, gall midges are there, stem borers are there, aphids are there, mites are there, flies are there. Good number of pests are seen attacking our standing crops and also the stored produce in the godowns. So here, what we do is, so not only insects, even small animals fall under what we call the term pests. So here, the disease is nothing but a disorder in the structure or function of the plant or a produce. So now here, of course, we discussed a lot about, so diseases are caused by, pathogens, that is living and also non-living. So here, non-living means the inclement weather, too hot, too cold would be there, or too much humidity will be there, and sunburn will be there. So these are all, they do not get transmitted. They are not infectious. Whereas the diseases caused by bacteria, viruses, or mycoplasma, they are transmittable. They get transmitted from one host to the other host. So now, when this disease or the concepts of principles of pest and disease management involves, if you look at the disease, generally there is what we call a disease triangle. What we call a disease triangle is there. In this disease triangle, you find what we call a host, a pathogen, and the environment. The host, pathogen, and the environment make what we call a disease triangle. So our aim should be, or the principle of this disease management or pest management involves breaking this triangle. Breaking this triangle or excluding at least any one of these conditions. If once the chain is broken, it is very easy for us to bring disease under control and to protect our crop. So now here the management, the management principles, the principles of management, generally, it was, in 1929, Wedgel in America, he proposed, he classified various principles of pest disease and pest management. Now, of course, the term management, that is principles, was given by, or the classification of various protective measures were classified by Wedgel, whereas the concept of disease management, pest and plant disease management, was Gear and Clark. Gear and Clark in 1961 proposed the term management. So here management means not one. We discuss various concepts. Various concepts are so based on where the disease is there, what type of crop you are growing, what type of environment is there, what type of soil is there. So based on this, you choose one of the management practices. So here management, it is a holistic approach wherein you use not only your avoidance, your exclusion, eradication, your protection, immunization, therapy. So these are all some of the principles of plant disease management given by Gear and Clark management, but Wedgel proposed this classification of management. So now here the process involves the prevention is better than cure. It is a common saying goes on. So after struggling, after getting infected and then struggling, going for therapy, the best thing is to protect yourself from the onslaught of the pathogen. See that you avoid coming in close contact with the pathogen. If you avoid that, you are safe. So here, the principles of plant disease management, the fundamental principle, this is very, very important as far as your exam is concerned. They'll ask you, write an essay on principles of plant disease management. So these principles, the classification of these principles were given by Wedgel in 1929. There are primarily 6 principles. What are they? Avoidance, exclusion, eradication, protection, immunization, and therapy. So we'll discuss one by one. First of all, avoidance. What is avoidance? Here, avoidance means an attempt to see that pathogen is kept at bay. That pathogen is not allowed to come in contact with host. This is avoidance. Avoidance, of course, if you remember our COVID-19, we took a number of measures, to avoid. What was the best thing? Wearing a mask, sanitization, keeping aloof, cleaning, sanitization, thorough sanitization. So this was avoiding coming in contact with the pathogen. This is what we call avoidance. So now here, as far as the crop is concerned, this avoidance involves, this involves selection of the proper field. Your field should be, your area where you're growing crop should be away from the inoculum. It should be away from the inoculum. The field you select. Say, if you grow in an area which is already infested by that pathogen, means you cannot protect your field. So your area should be quite far from the area which is already has the inoculum in that area or disease causing pathogen in that area. So avoiding. So this involves selecting a geographical area and selecting a proper field and also planting time. Now, you might have seen, yesterday our CM was in an interaction with the IAS and agricultural scientists. He said that let us prepone the sowing time so that, now we have seen, now the produce is ready for harvest. Even harvest of paddy has taken place. But because of unusual rains, unexpected rains, lot of produce is lost. Farmers are put to great losses. So now to counter this, what you have to do? You have to prepone, go for early sowing. If you go for early sowing, by the end of April, if you are able to reap your crop, generally we have seen, for the last 20 days, we are experiencing rains. Now this is the time, where unusual rains are expected, hailstorms are expected, lightnings are expected. So if you prepone, that is, if you go for sowing instead of October, if you start in the month of September, your crop comes for harvest by the it is second or third week of April. Then your crop will be safe. So here avoidance. So here a selection of the proper field, selection of the proper geographical area, and planting time. That is, go for early plantation. And the other is go for selecting disease resistant or pest resistant varieties. Even if you even if a pest comes, then also if your crop has proper immune system, so the pathogen will not be in a position to make an entry into the, or if it tries, even if it enters also, it cannot reproduce and cause a lot of damage to your crop. So here the not only avoidance of the pathogen coming in contact with the host. So for this, select a proper geographical area and go for the proper field and go for the proper plantation time and go for disease resistant varieties, and also see that that area is free from insects, that is, which are vectors that carry the pathogens. Then also see that proper sanitation is there in your area. Proper sanitation means these are rogues or the weeds. The weeds are which form the alternate hosts. Their population should be checked. This is what we call avoidance. Now, for example, there are certain examples are there. For example, proper timing, timing should be there, I told you. So bean anthracnose, this is caused by what we call Colletotrichum lindemuthianum, is the fungus imperfecti, fusarium species or Colletotrichum species that cause, anthracnose disease on beans. Now it has been an experience that if there is a prolonged rain, the disease spreads very fast. So you should go for plantation in such a way that your crop comes for harvest before the heavy rains in your particular area. See, the rains you get, whatever the rains you get in Telangana, so generally we get southwest monsoon. If you go to Andhra areas or Nellore areas or Rayalaseema areas, even not only southwest, even northeast monsoon is also very active there. So see that this prolonged rainfall will not be there in your area, when your crop comes for harvest. So this is what we call avoidance. So even another example is there. For example, in case of bacterial blight of bean also, in addition to this bean anthracnose, bean also suffers from what we call bacterial blight. Now this blight is caused by Xanthomonas phaseoli or Pseudomonas phaseoli. So they cause what we call blight in bean. So now this bean, it has been proved that the bean blight, bacterial blight of bean is a seed borne, a bacterial. The bacteria are present on the seed coat. So as it is seed borne, if you take proper precautions or see that your material or seed material is free of pathogen, then you are safe. Your crop is safe. So what you do, you treat your seed. Use various seed treating materials, either chemicals or sulfur dust, or your Cerazon, Agrasone, etc. Or you go for what we call hot water treatment. By doing all these things, if the pathogen is a seed borne, then you can get rid of that pathogen from your seed If such treated seeds you sow, then your crop will be safe. So also, even for example, in case of potatoes suffer from what we call a virus disease. So go for the seed material. So here in potatoes, the seed material is not our embryonic seed. Here, this is a small tuber, a tuber with eyes that forms your seed material. So get your seed from an area which is not infested from these diseases. So here what I mean to say is select the seed, proper seed, disease-free seed, go for early sowing, select an area which is free from or that infested areas. So this is how you can avoid the onslaught of the pathogen onto your crop. This is what we call avoidance, simply avoiding. Avoiding contact with the host and the pathogen is avoidance. Then the second principle is what we call exclusion. Now, the exclusion means here it is a what we call, this involves preventing. Exclusion is preventing the entrance and the establishment of the pathogen in an uninfected area is what we call exclusion. So exclusion is preventing the entrance and the establishment of a given pathogen in an area which is uninfected so far. That is what we call exclusion. So the best thing is what we call quarantine. Again, quarantine. This was a famous term during our COVID times, quarantine. So especially when foreigners used to come, soon after their landing in an airport, they were taken to a facility where thorough check was there. And generally, if the people coming from infested areas or COVID affected areas, then the passengers were supposed to, it was mandatory for them to keep aloof, to keep in a given room for 3 or 4 days, then pre-check and the post-check. Then after confirmation from the medical expert, then only they will be allowed to come out and mingle with other people. So this is what we call quarantine. So what is quarantine? It is a legal restriction to prevent and the entrance and the establishment of the disease or an insect into an area which is uninfected. This is what we call quarantine. So it is a legal restriction. Legal means enforced by the police or the law officers. You will not be allowed to mingle with, to enter into an area until you are given, a NOC by that respective medical officer. So here also quarantine. So here what you mean here quarantine means, see so many seed materials, maybe your flower, your tubers, or your rhizomes, or seed, etc. For growing and also for the research purpose, we import from various other countries. So while importing, what we have to do, agricultural scientists, they should check that at the airports or at the seaports or on the borders. These agricultural scientists should check that material, whether it has any pathogen, whether it is seed-borne, whether that is present in the flowers or the tubers you are importing. So they have to be checked, thoroughly checked for the infestation with any pathogen. This is what we call quarantine. So the scientists, the agricultural scientists or the agricultural officers, they check the material, they inspect the material. So here exclusion involves 3 or 4 important processes. One is quarantine, inspection, certification, and the last one is treatment, seed treatment. So quarantine is a legal binding. Then inspection means the qualified persons will be checking, inspecting the material, seed or the other vegetable material, whether it is disease-free or the pathogen-free. This is what we call inspection. Then the third one is what we call certification. They'll give you seed. See, now even our government is, asking the farmers to go for only certified seeds, treated seeds. Say if you go for, say when in another 20 days or so, when the monsoon will be active, then farmers start, sowing season. Then they go for various, these fertilizer and seed selling market or shops. There they will get the certified seed. Certified seed means it is pre-checked and certified that it has no pathogen. It has good viable seeds. It is free from any pathogen. So it is safe to grow them. So this is the guarantee given by the experts. We call such seeds as certified seeds. Then seed treatment, I already told you. So if you have any suspicion, if you're suspicious, what you have to do? You go for seed treatment. Various chemicals are there. Hot water treatment is there. Agroson, Cerason is there. Sulfur dust is there. By using these materials, you can make your seed pathogen free. So this exclusion is what we call it. It tries to restrict the entry of the pathogen and its establishment in an area which is so far not infected by that pathogen. This is what we call exclusion. Okay, now in India, of course, say in USA, example is there. For example, in 1910, American people imported what we call, lemon, nimbu. They imported, and in their orchards, they saw the citrus canker. Citrus canker, Xanthomonas axinopodus citri, So it causes citrus canker. So all the citrus orchards were affected by this. So government of America has taken steps that they cut all the infected or all the orchards, they burnt them and made within a few months, all their orchards were free from the citrus canker. So now even in India also, we have what we call Destructive Insect and Pest Act, DIP Act, what we call, that was enacted in 1914. Of course, this after independence, of course, we have, Ministry of Agriculture and Indian Agricultural Research Institute is there. And of course, we have what we call NBPGR, this National Bureau of Plant Genetic Research. Now, this is a nodal agency that supervises. If you want to import seed material, whether it is your embryonic seed or your flowers or the seed material, vegetable seed material or vegetative seed like tubers, etc., they are to be prior to the import. You have to take the permission of the NBPGR. So NBPGR gives you, a license to import that. After reaching the destination, especially airport or seaport, our people, especially agriculture scientists, again they check it, they inspect it, they'll certify it. Then only that will be allowed to be used in a particular area. So this exclusion involves what we call legalized or regulatory provision aiming at checking the entry of the pathogen into an area which is not so far infected by that pathogen. This is the second principle. Third one is, third pest management principle is what we call eradication. Now, the eradication means here what we do, once, of course, eradication, you have taken so many steps. First of all, what did you do? You avoided. But instead of avoiding also, if the pathogen makes an entry into your crop or into your storage area, what you have to do? You have to eradicate it. Eradication involves what we call sanitization measures or cultural methods or what we call agronomic methods. All these are aimed at reducing the dose of the inoculum in that particular area at a minimum level. So here we see that the inoculum is present at barest minimum. So eradication, see, the control. Eradication is complete removal. See, we have no right to kill anything, to remove a particular individual from the surface of the earth. Our aim should be to check their population, to keep their population in such a way that they will not be causing harm to our produce. See, once you eliminate, once you destroy a given species, means you have no power to regenerate them. Once lost means lost. So our aim should be to keep their population under check. See, your crop cannot be you and me. We cannot remain, uninfected by any pathogen. It's also not good. Unless and until you are infected with some pathogen, your immune system will be at idle. So if any pathogen, any disease-causing organism enters your body, then your immune system wakes up and it tries, it gets ready and to fight. So here also, of course, crop is susceptible for a disease. But see that pathogen will not cause harm to your crop in such a that 1 or 2% loss will be nominal. But if the loss exceeds 20 or 30%, then it is alarming. So here, what we do in eradication is you remove the infected parts, the diseased parts, stems, leaves, etc. They should be cut. They should be burnt. You go for roguing, that is the alternate hosts that harbor when crop is not present, then the pathogen survives in the collateral host. So see that your field is free from these alternate hosts or the rogues, what we call. Then go for what we call crop rotation. See, if you grow same crop year after year, if your crop gets affected with a pathogen, means if you grow, say, again and again same crop, the volume of the pathogen gets increased year after year. So then you'll put to great loss. So see that you go for crop rotation. See, a pathogen that causes, say for example, Sphaerotheca sorghum that causes, covered smut. It will not be in a Sphaerotheca cannot cause the same disease in rice or same disease in wheat. So each pathogen has what we call host to specificity. Unless and until it gets that particular host, it will not be in a position to cause any disease. So hence, if your crop is affected, means don't go for the same crop next year. Go for what we call rotation. If you are growing, say for example, if you grow sorghum this year, then go for next year grow your arhar, what we call red gram. Then you go for cotton. Then you go for what we call pearl millet. So like this, change every year. For your first crop should get its its line only after 4 or 5 years. So this is what we call crop rotation. Then of course what we call disinfection by heating. Disinfection by heating, see what we call soil solarization. So if say pathogens, all our pathogen, maybe insect eggs, your larvae, pupae are pathogens, fungal or bacterial or viral or mycoplasma. They cause disease, and when you reap, the crop tubules or the fallen leaves, they remain in the field. So they form the fresh source of inoculum for the next crop. So then, hence, what you have to do, you have to see that your soil is free from. So you go for what we call soil solarization, or you go for deep ploughing of the soil, or you treat your soil with certain chemical so that the tubules or the pathogen or the alternate that is surviving on the alternate host, they are removed. And once your field is free from, or if it is fully sanitized, then you can go for the raising of the second crop. So this what we call sanitation can be done by using soil solarization, but heat, that is heating, or by spraying certain chemicals, say, or removing a certain alternate source. Say we read about what we call wheat rust or black stem rust of wheat. Puccinia graminis triticea we have seen. It is a macrocyclic that requires two hosts we have seen. The wheat is the primary host and the barberry is the secondary host. So for the completion of the life cycle, what is needed? The pathogen needs the alternate host. So if you see that the barberry is not there in the area, then Puccinia cannot complete its life cycle. So your crop is safe. So this is what we call, eradication. So avoidance, then exclusion, then eradication is there, then protection. What do you mean by this protection? Protecting your crop by various methods. Say, for example, by growing certain plants. If there is a gust of wind, along the wind, your pathogen or the bacteria or the fungal spores, they travel. So if you erect certain windbreaks are there, if you erect certain what we call sticky traps. Sticky traps are there, or if you spray certain repellents. Say, for example, good example for this is plant product. For example, if your area has what we call mosquito menace, then what you do? You go for what we call odomas. So it acts as a repellent. By sensing, by smelling that, the mosquito, it acts as a mosquito repellent. So here also you go for what we call repellents in the form of windbreaks or mechanical barriers, etc. By using this, generally by spraying certain fungicides or erecting windy breaks or mechanical barriers, you can protect your crops. And the fifth one is what we call immunization. Immunization means going for disease-resistant varieties. So, agricultural scientists, especially plant breeders, by using various this breeding techniques, they introduce these resistant genes from the wild ones to our cultivars, thereby enhance this immunization. Immunization is nothing but ability to face the onslaught of the pathogen. Even if pathogen attacks, then you will remain, only partially damaged. So that is what we call immunization. So this immunization is affected by breeders, plant breeders. A number of varieties are now available which are resistant to various fungal diseases, bacterial or viral diseases. So disease-resistant varieties, for example, cereal rust, we have powdery mildews, downy mildew resistance are available with us. Now, especially what we call transgenic plants. Of course, we'll discuss about them in the later classes. So by using what we call rDNA technology, recently these transgenic plants are produced, and a good example for that is BT cotton, BT brinjal, etc. Even if an aphid comes, it eats also, it dies. You need not spray any chemical. This is possible by what we call disease-resistant varieties. And the last one is what we call therapy. Therapy means even after taking almost all the precautions, even then if your crop suffers one, what you have to do? You go for therapy. That is using certain chemicals or by using what we call thermotherapy, chemotherapy. These are the two therapies that are helpful for us in protecting your crop. So this is what we call various principles that help you to protect your crop. So here we discussed about what is avoidance, what is exclusion, what is eradication, what is protection, what is immunization, what is therapy. So all these form what we call various principles of plant disease management. Then we'll move to, of course, the same thing, little bit elaboration now, one after the other. See, various methods of plant protection. See, this pest management involves primarily preventive measures and the curative measures. The pest management involves two processes. One is what we call preventive measures. The other is what we call curative measures. Preventive means prevention is always better than cure. Prevention is always better than cure. So now this prevention involves, this preventive measures involve what we call cultural, your mechanical, your physical, your genetic, your regulatory, and immunity. These 5 fall under what we call prophylaxis or preventive measures. Even after taking preventive measures, if your crop suffers from a disease, then you go for, as a last resort, you go for curative. Curative measures are primarily of two types. One is what we call biological method, and the last one is the chemical methods. See, these are all measures to check the spread of the disease, which aim at to keeping the dose of the inoculum at bare minimum, so that your crop doesn't suffer from severe losses. Now, the preventive measures. So, or prophylactic measures, preventive measures. So, our aim is to prevent the entry of the pathogen into your field or into your go down. That is a prevention. Even after taking so many preventions, even then if your crop suffers from a disease, then you go for what we call curation. Say, we take ourselves. You go on, you take, daily bath, you go for, you change your lifestyle, you take proper nutritive food, you take, proper sleep, then you'll be healthy. But even after taking so many measures, there is no guarantee that you don't suffer from any disease. Even after taking so many precautions, if you suffer from a disease, then you go for curation, that curative. So preventive measures and curative measures. So here there are two principles. One is prevention and the cure. So I told you, so generally it was Geer and Clark who first proposed the term pest management. Of course, the classification of various pest management were given by, as I already told you. So it was Wedgell who classified them into preventive measures and curative measures. So now we'll discuss one after the other. So first one is preventive measures. First preventive measure is cultural method, or what we call agronomic practice. Agronomic practice are, so here, as the name itself tells you, farmer knowingly or unknowingly, intentionally or unintentionally, this is the season, farmers go for thorough ploughing of their field. The ploughing is taken up with two aims. One is to loosen the soil, to bring the deep soil onto the top. While doing so, the pathogens or the previous crop stubules that form the source for the next crop pathogen, they are brought onto the top, and the eggs of the larvae or pupa, which are hidden inside the soil, when you plow it, they come onto the surface, and when they are exposed to hot sun, they get killed. So this the cultural agronomic practices help us, to kill, to check, to keep the pathogen at minimal level. So what are the various things? Generally you go for what we call thorough ploughing of the field, wherein you bring egg, larvae, pupae onto the, and also the soil inhabiting various insects such as wiry worms can be brought onto the surface, and the birds which are there, they spot them and they kill them, they eat them. Second one is what we call cultural or agronomic practice, is going for early and also synchronized planting. All in your area, they are to be planted at a given time. If somebody plants, after 15 days, another after 1 month, so if the early planted one at the end of the season may develop certain disease, the pathogen gets transmitted to the next field, then to the next field. So to avoid this, if you go for synchronizing it or at a time, at an optimal time of planting has to be taken up so that your crop is protected. This is what we call synchronized planting. Then, of course, intercropping, wherein not one, 2 or 3 crops you plant at a time, so that the pathogen gets confused. The beetles or the worms or your leaf suckers, sap suckers, borers, etc. They get confused. Which is susceptible, which is non-susceptible. So if you are growing 2 or 3 crops at a time, mixed cropping, some are susceptible, some are non-susceptible. So thereby even the susceptibles, they get protected. This is one. Then crop rotation, of course, we discussed it. Then what we call pruning and thinning. Pruning and thinning is nothing. Pruning and thinning, in certain cases, what you do? You prune it. You remove the top, tipping. If say generally pruning, generally we take up even in orchards, generally diseased parts, diseased branches, diseased leaves, etc. They are cut and they are burnt. This is what we call pruning. Pruning helps, while cutting, what helps? It helps us for aeration, and your light will be there. So the humidity will be checked. If a lot of humidity is there, this is congenial for the spread of the fungal diseases. So hence pruning has to be there, done so that penetration of the light will be there, good aeration will be there, so that spreading of the pathogen can be checked. Then, of course, what we call management, water management should be there, and even fertilizer management should be there. So your fertilizers, generally people in our area, because of little knowledge, illiteracy, they think that if you give more fertilizer, they think that you get more yield, but that's not so. If your doses of nitrogen is more, generally your crop, you are prone for disease or pest attack. So the NPK should be given in a judicious way or in split doses. Okay, then this crop residues, so they should be, destruction should be there, and eradication of the harmful and the alternate hosts should be there. For example, in case of rice, it is panicum, it is Citeria, Cynodon, Echinocloa. These are some of the alternate hosts growing along with the hedges or along the boundaries of your rice crops. So they are to be eradicated. They are to be removed. Your water management should be there. So if water is sufficient, then you flood your field. Flood your field. If flooding, then the pupae or the eggs that are there, the insects, they get washed away. Then you see that your field is exposed to hot sun. So alternately, wet and dry conditions. If you expose that, then the pathogen will not be in a position to survive. This is one. And then already told you, cleaning the fields and the borders, edges with your orchards also, your dropped fruit. So if you go to orchards, orchards means fruit yielding, mangoes or your what we call citrus fruits, etc. are there, your apples are there. The fallen fruits, they have, on the if you do not remove them, fallen fruits, you will see insects breeding there, your pathogens breeding there. So it forms the inoculum for the next cycle of disease. Then what we call second will be what we call, first is the agronomic or what we call cultural. Second method will be what we call mechanical method. So here the mechanical method means, here a mechanical means pulling and cutting the diseased parts. So hand destruction, using your hand, using implements, cutting them, burying them, burning them is what you call mechanical. So then exclusion of barriers. You erect certain barriers. You grow certain plants. Say, for example, in case of beans, if you are growing in pepper gardens, you are growing there. Or, for example, your groundnut is there, very, very herbaceous. In that areas, if you grow, certain tall varieties such as sorghum or pearl-minted elytra, or even what we call, castor, etc., on the hedges or on the borders, they act as barriers. The insects, they coming, they feed on these borders or the leaves that are the plant that you have raised on the borders, the insects feed on them. So these aphids, they carry certain, viruses, etc. So the virus cannot live long. So by the time it feeds on there, by the time it reaches your actual, your crop, by the time, the virus that is there inside, it gets gut, it gets killed, it gets inactivated, that pathogen. So like this, in certain cases, certain wheat is a hollow stem. So here, certain flies and leaf hoppers which feed on wheat, where the stem borers, or even rice also. The culm is very spongy, penetrable. In that areas, if you grow solid stemmed plants, the insect comes, of course, it lays its eggs, but its grubs or its larvae will not be in a position to enter into the solid stems. Thereby, they get killed. So this is how the soft stemmed and the solid stemmed ones, if you grow together, generally your crop gets protected. Then, of course, by erecting what we call certain or growing certain crops, such as, for example, crotalaria is there. For example, Solanum nigrum. These are what we call crop traps. See, in certain areas, generally the nematode, a root knot nematode, Meridogyne. So generally it is found, generally in the tobacco growing areas, it feeds on the root system, and it develops what root knots. And of course, the root system gets destroyed, thereby your produce, that is even your leaves, they droop, they dry, they do not develop properly. So to check them, if you grow along with your tobacco crop, if you grow crotalaria, Janum, what we call, or Solanum nigrum, it has been proved that their root system exudes certain chemicals that attract the nematodes. So to lay their eggs there, and they hatch their eggs, and they will not be, the young ones will not be able to penetrate into the root system of your Solanum nigrum or Crotalaria gentium. So they get killed. So such crops which protect your susceptible crop, acting as crop traps. Trap ante into utsu. Utsu padipotadi. So crop traps. So such ones are known as crop traps. And of course, even we have, certain marigold or asparagus, they also act as antagonistic, inimical to nematodes. Then, of course, physical methods. The third one is physical methods. Physical means here environmental, mainly high temperature or low temperature, or air, or what we call a light or radiation. By using these physical energies, that is subjecting your field, subjecting your soil to high temperature or low temperature, or what we call exposing, or in the hedges or in the borders, if you erect certain traps, certain films, say sticky films, certain sticky films are there, maybe you are transmitting or glowing, maybe yellow ones are there, aluminum foil like are there. They reflect light on the incoming insects. So they fly away, they move away. Or certain insects will come and they stick to the traps you have raised in that area. Or else you can what we call light traps. In the evenings, generally this is a common practice. So generally by the evening time, generally farmers raise fires on the hedges. So the insects which were sleepy, so they become active by evening, and looking at the fire, they get attracted to the fire and they fall in the fire and they get died. So either fires or sticky traps or reflecting traps, etc., or by blowing hot air or by spreading what we call polythene sheets. See, if you spread a polythene sheet on a moist soil, if you keep it for 2 or 3 days, it has been proved that on the sheet, below the sheet, generally, if the temperature around you is 37 degrees, below your the polythene sheet, the temperature will be around 50 degrees centigrade or so. So under high temperature, the eggs, pupa, larvae, or the spores of pathogens, the spores of your bacteria or even fungal spores, they get killed at hot temperature or at different wavelength radiations. Of course, soil solarization, that is, then of course, exposing your seed. See, this is a common practice, This is a common practice before sowing, Of course, if you are going for a market, if you going for, certified seed, of course, then it's okay. It's already treated. But if you use your produce as a seed for the next crop, then you go for what we call seed treatment. So this has been proved, especially hot water treatment in case of, smut of sorghum caused by Sphaerotheca sorghum. Now, is disease borne. So seed borne disease or your chlamydospores or your fungal spores are seen. This black sooty is seen on the surface of the seeds. So what you do, you keep the seed in hot water of 52 degrees centigrade for 10 minutes. Then the pathogen, the fungal spore, it germinates. And later on, after 10 minutes, you throw it, you spread it on the sunlight or in the shade. The fungal spore that produces, mycelium, it germinates. That will not be in a position to tolerate this high temperature. It dies. Or you can treat your seed with sulfur, cerazone, pyrom, etc. By using that, you can check the disease seed-borne pathogens. So under physical, already told you, application of the heat is there. Then soil solarization, already spreading of the plastic sheets, transparent plastic sheets is there. Then exposing the infested seed to hot summer or hot water is there. Then of course the light traps already we discussed. Then of course at night, generally flying insects will come, get attracted to the light, and they get killed. Of course, even not only that, generally bugs, cutworms, etc., leafhoppers, etc., they get attracted to these light traps or the sticky traps and they get killed. So in certain cases, of course, you can make your seed, by exposing to radiation, your ultraviolet rate. So if you expose your seed to ultraviolet radiation, ultraviolet radiation kills the pathogens. So then your seed becomes pathogen free or spore free. So then you can go for what we call, you go ahead using that seed as your sowing seed for the next crop. So this is about so we discussed in today's class about various principles involved in pest management. Various principles involved in pest management we have seen. Those principles were what we call, avoidance was there, and exclusion was there, eradication was there, then protection was there, then immunization was there, and the last one was therapy. These 6 form what we call principles of plant disease management. Then of course, these management, the classification, of course, is primarily into 2. One is what we call preventive measures, and the other is curative measures. Under preventive measures, we have seen What are the cultural methods? Cultural means agronomic. Agronomic various practices such as ploughing and intercropping, mixed cropping, crop rotation, and a removal of your, rogue plants, uprooting of your previous crops, tubules, etc. is what we call cultural. Of course, mechanical we have seen, erecting physical barriers, etc. And also you just now we have seen various mechanical, various traps, etc. Sticky traps or reflecting traps, etc. were there. And of course, the other was what we call the other measure was physical, wherein the that involves, physical means environmental, that is hot air or cold air or what we call radiation and light traps, etc. These are various practices. See, what you have to understand ultimately is that, see, you need, say, for each crop, for each pathogen, these measures, your preventive measures or curative measures, differ from crop to crop, area to area, geographical area to area, or the nature of the soil, etc., nature of the environment, etc. They differ. So hence, these are all collective measures. So you have to choose. After identifying the pathogen, you have to choose which preventive measure or which curative measure is suitable for your crop. So this is about principles and the principles of your pest and disease management. And if you have any doubt, please raise your doubts so that I'll try to answer them. If you have no doubt, then I'll close my lecture for today. And as you know, we have one more class tomorrow and also day after tomorrow. So try to attend these 2 classes because you have very little time before you take your exam. And if you attend to these online classes, then you'll be saving a lot of time. So this will help you, to understand better. That is the idea of arranging these online classes. So if you have no doubt, then I'll say goodbye and let us meet tomorrow, same time, 12 to 1 p.m. Thank you.